Welcome back, everyone, to another deep dive. This time we're going microscopic, getting into some fascinating stuff about cellular injury.
Really interesting topic.
Yeah, and you guys sent us some really interesting material to work with this time. We have some academic lecture notes in Portuguese.
A challenge.
Yeah, a bit of a challenge. Some medical illustrations and diagrams.
Mhm.
So we're going to put all this together and really try to uncover how cells respond to stress. What actually causes this injury, and then how do the medical pros study it,
Yeah.
and get this, cells can actually commit suicide. It's called apoptosis, and it's way more interesting than it sounds.
It's fascinating. We think of cells as, these simple building blocks, but really when you get down to it, they're incredibly complex systems. And when those systems are pushed too far by stress, by injury, that's when we start to see the
I mean,
problems.
this is a cellular injury. It's like pushing a cell to its breaking point.
Exactly. It's that disruption of the cell's structure and function. Now, the good news is this can be reversible, meaning the cell can bounce back. But if the damage is severe enough, it becomes irreversible and the cell dies.
So it's not just a binary dead or alive situation. There's a whole spectrum of injury.
Absolutely. And what's more, different parts of the cell, like the mitochondria, the powerhouses, can be specifically affected.
I'm curious, what determines whether a cell can recover or not?
It really depends on the severity and the duration of the stress. It's key factors. Think of it like stretching a rubber band, right?
Okay.
A little stretch, it snaps back.
Right.
But stretch it too far for too long and it breaks.
Yeah.
Cells are very similar. Mild temporary stress, they might be able to repair themselves.
Okay.
But intense or prolonged stress, that's when the damage becomes irreversible.
Oh, that's a great analogy. Okay, so let's say a cell is experiencing reversible injury, that milder stress. How does it respond initially? What happens?
So two key things happen. First, energy production takes a hit. Cells use a molecule called ATP for energy, and stress hampers their ability to make it. It's like running out of fuel.
Uh-huh.
The second thing is the cell starts to swell. This happens because it struggles to maintain the balance of ions and water inside. Imagine your body struggling to keep everything in equilibrium.
So it's all about maintaining that really delicate balance. It's like a microscopic tightrope walk.
Exactly. But what happens when that balance is disrupted beyond repair? Well, that's when we start talking about cell death.
Okay, you mentioned that cell death can be either necrosis or apoptosis. Necrosis sounds pretty grim, like a building collapsing.
It is. Necrosis is messy and uncontrolled, and it's always pathological, meaning it's a sign of disease. Imagine a cell bursting open, spilling its contents, and triggering inflammation. Not good.
Yikes, that's a vivid image. And apoptosis, you said, is different.
Very different. Apoptosis is programmed cell death. It's like a controlled demolition. Neat, orderly, and often beneficial.
So the cell basically self-destructs. Why would it do that?
Well, there's a few reasons. During development, apoptosis helps sculpt our tissues and organs by removing unnecessary cells. Think of it like sculpting a statue. You'd chip away the excess material to reveal the final form.
Okay.
It also gets rid of cells that are infected with viruses or have damaged DNA, preventing them from replicating and causing harm. Apoptosis is essential for maintaining the overall health of the body.
So it's not always a bad thing. Sometimes cell death is actually a good thing.
Exactly. It's all about maintaining balance and ensuring the well-being of the organism as a whole.
This is fascinating.
Okay.
Okay, let's shift gears a bit. What are some of the things that can actually cause cellular injury? What are the usual suspects?
Well, there's a whole rogues gallery of threats to our microscopic world. We can kind of group them into categories to make it easier to understand. First up, we have oxygen deprivation.
Oxygen deprivation. That sounds serious. Tell me more.
So our cells need a constant supply of oxygen to function. When they don't get enough, it's called hypoxia.
Okay.
And in severe cases, complete oxygen deprivation is called anoxia. And think back to energy production without oxygen. Cells can't generate enough ATP and their functions start to shut down.
So it's like a power outage for the cells.
Exactly. One major example is ischemia. That's a reduction in blood flow to a part of the body, reducing the supply of oxygen and nutrients. Ischemia is a key player in conditions like heart attacks and strokes, where blood clots block blood vessels. And the scary part is epidemiological studies show that ischemia affects about 7% of men over 50.
7%. That's a lot higher than I would have guessed. So anything that restricts blood flow can lead to ischemia and potentially cellular injury.
Precisely. And it's important to remember that ischemia can be chronic, meaning it happens over time, or acute, meaning it's sudden and severe. Chronic ischemia of the lower extremities, for instance, can lead to pain, ulcers, and even amputation. Acute ischemia, as in a heart attack, can be life-threatening. It really highlights how critical proper blood flow is for cellular health.
Yeah, that's a sobering thought. Speaking of critical factors, what about nutrition? What role does it play in cellular injury?
Nutrition is absolutely fundamental. Just like we need the right nutrients to thrive, so do our cells. Nutritional imbalances, whether it's deficiencies or excesses, can really wreak havoc on cellular health.
So it's not just about maintaining a healthy weight. It's about giving our cells the building blocks they need to function.
Exactly. Take a deficiency like anorexia. When the body is starved of essential nutrients, cells can't perform their tasks properly, leading to widespread dysfunction. On the flip side, excesses like those seen in obesity and diabetes can also be damaging. High levels of glucose and fats in the blood can be toxic to cells, leading to inflammation and oxidative stress.
It's all about balance, right? Too much or too little of anything can throw things off.
Precisely. But it's not just about what we put into our bodies. External factors can also play a role. For instance, we have physical agents like trauma, burns, radiation, extreme temperatures, and pressure changes.
Yeah, I could see how those would be problematic. A physical blow could crush cells and extreme heat could cook them. Right.
Exactly. And radiation can damage DNA, leading to mutations and cell death.
So cells are pretty vulnerable to these physical insults. What about chemical agents? I imagine there's a wide range of those that can cause harm.
Oh, you're absolutely right. We encounter them every day.
Wow.
Even something as simple as water. In excessive amounts can disrupt the balance of electrolytes in our cells, causing swelling and dysfunction. Glucose and salt, essential in the right amounts, become harmful in excess. Then we have toxins like arsenic and cyanide, which can directly interfere with cellular processes, leading to rapid cell death.
It's a reminder that the dose makes the poison, as they say. What about environmental pollutants? Do those play a role too?
They absolutely do. Air and water pollution, industrial chemicals, pesticides, all of these can damage our cells. And here's a fascinating fact. Even oxygen, essential for life, can become harmful in high concentrations. It's a reminder that balance is key.
Wow, I never thought of oxygen as a potential threat. Okay, so we talked about physical and chemical agents. What about biological threats like bacteria and viruses? How do those cause cellular injury?
Infectious agents, including bacteria, viruses, fungi, and parasites, have evolved various ways to attack our cells. Some release toxins that directly damage cells, while others hijack cellular machinery to replicate themselves, ultimately destroying the host cell.
So it's like a microscopic battleground going on inside us. But isn't our immune system supposed to protect us from these invaders?
It is, and it does an incredible job most of the time. But the immune system is a powerful weapon, and sometimes it can turn against our own cells, causing what we call autoimmune diseases.
So in these cases, our own defense system becomes the attacker.
Yeah, it's a bit of a double-edged sword. In autoimmune diseases, the immune system mistakenly identifies our own cells as foreign invaders and attacks them. This can lead to a wide range of conditions, from rheumatoid arthritis, where the immune system attacks the joints, to lupus, which affects multiple organs.
It's fascinating and a little bit scary how complex and powerful our immune system is. Are there any other causes of cellular injury we should be aware of?
Yes. We also have genetic damage to consider. Our DNA contains the blueprints for all cellular functions, and any errors in these blueprints can disrupt crucial processes, leading to disease. These errors can be inherited or acquired over time due to environmental factors like radiation.
So genetic damage is like a glitch in the cellular software.
That's a great way to put it. And these glitches can have wide-ranging effects, from mild dysfunction to severe disease. Some people are even genetically predisposed to certain diseases because of specific gene variants they inherit. It's why some people are more susceptible to certain illnesses than others.
That makes sense. Genetics play a huge role in our health. What about aging? We all know our bodies change as we get older. Does aging contribute to cellular injury?
It absolutely does. Aging is a natural process that involves a gradual decline in cell function and repair capacity. As we age, our cells accumulate damage, and their ability to repair themselves diminishes. It's like a car that's been driven for many years. It starts to show wear and tear.
So aging is kind of like a slow, cumulative form of cellular injury.
You could say that. There are even specific structures in our cells called telomeres that play a role in aging. Telomeres are like caps on the ends of our chromosomes, protecting them from damage. Each time a cell divides, its telomeres get a bit shorter. Eventually, they become so short that the cell can no longer divide and dies. This process is thought to be a key contributor to the aging process.
Wow, it's incredible how much is happening at the cellular level, even as we age. So we've covered a lot of ground here: oxygen deprivation, physical trauma, chemical exposure, infections, autoimmune diseases, genetic damage, and aging. But how do we actually study cellular injury? How do doctors and scientists figure out what's going on at such a microscopic level?
Well, that's where the field of anatomical pathology comes in. Anatomical pathology is all about examining tissues to understand disease. It's like detective work, looking for clues at the cellular level to diagnose and understand various conditions.
Okay, so anatomical pathologists are like the detectives of the medical world. What tools do they use to investigate cellular injury?
One of their key tools is the biopsy. A biopsy is a procedure where a small sample of tissue is removed from the body and examined under a microscope. And there are different types of biopsies depending on the location and type of tissue being examined.
So it's like taking a tiny snapshot of what's happening inside the body. What are they looking for in those snapshots?
It's all about those telltale signs, those subtle changes in the cells that point to injury or disease. For example, reversible cell injury might show up as cellular swelling. Under the microscope, the cells look enlarged with pale cytoplasm.
So it's not just about seeing if cells are dead or alive. It's about looking for these more nuanced visual clues. What other changes might indicate injury?
Exactly. We might see fatty changes where fat droplets build up inside the cell, making it look foamy under the microscope. This often happens in liver cells when they're stressed.
Fatty liver. That makes sense, given the liver's role in fat metabolism. What other visual cues do pathologists look for?
In more severe cases, we might see changes in the cell's nucleus. The chromatin might clump together or even fragment, which suggests the cell's DNA has been damaged.
So if the nucleus is messed up, does that mean the cell is beyond repair?
It's a strong indicator of irreversible injury. The nucleus is the control center of the cell, after all. If it's damaged, the cell's ability to function and repair itself is severely compromised.
That makes sense. You've also provided some information on how these tissue samples are prepped for examination. It sounds like quite a process.
It is. It's a meticulous multi-step procedure. Remember, we're dealing with incredibly thin slices of tissue, sometimes only a few cells thick, mounted on a glass slide.
It's mind-boggling how they managed to slice something that thin.
It really is impressive.
Yeah.
The process starts with fixing the tissue, preserving its structure using chemicals like formalin. Then, for hard tissues like bone, we need to remove the calcium through decalcification to make it soft enough to cut. After that, the tissue is embedded in a material like paraffin wax to provide support during slicing.
So it's like encasing the tissue in a tiny block of wax.
Precisely. And this block is then sliced into those incredibly thin sections using a specialized instrument called a microtome.
A microtome. It sounds like something out of a sci-fi movie. What about staining? You mentioned that earlier. What's the purpose of that?
Staining is crucial because most cells are naturally transparent. By using different dyes, we can highlight specific cellular structures, making them stand out under the microscope. It's like adding color to a black and white photo, bringing out those hidden details.
That's a great analogy. So by fixing, embedding, slicing, and staining, pathologists create detailed microscopic maps of the tissue, revealing the secrets hidden within the cells.
That's a beautiful way to describe it. These maps, along with the pathologist's expertise, help us diagnose diseases, understand how they progress, and even guide treatment decisions.
It's like having a window into the microscopic world of our bodies. We touched on the different types of biopsies earlier. Could you break those down a bit more for me?
Of course. Let's start with exfoliative biopsies. These are the least invasive, involving simply scraping cells from a surface. A good example is a pap smear, where cells are collected from the cervix to screen for cervical cancer.
Yeah, that's a fairly common procedure, so it's good to understand how it works. What about fine needle aspirations?
Fine needle aspiration biopsies, or FNAs, use a very thin needle to extract cells from a lump or suspicious area, often a tumor. It's commonly used to diagnose breast cancer, thyroid nodules, and lymph node enlargements.
So it's a way to get a sample of cells without a more invasive surgical procedure.
Exactly. And it's often guided by ultrasound or other imaging techniques to ensure they're targeting the right spot.
That makes sense. You also mentioned punch biopsies. When is that type of biopsy used?
Punch biopsies are used to take a small cylindrical core of tissue from the skin or other superficial areas. They're often used to diagnose skin conditions like rashes, infections, or suspicious moles.
So it's like a tiny cookie cutter for the skin.
That's a great analogy. And because it goes deeper than a simple scraping, it provides more information about the different layers of the skin.
Okay, that makes sense. You also mentioned incisional and excisional biopsies. What's the difference between those two?
An incisional biopsy removes only a portion of the lesion, while an excisional biopsy removes the entire lesion.
So with an excisional biopsy, the goal is to remove all of the abnormal tissue.
Exactly. They're often used for smaller lesions where complete removal is feasible and desirable, like for suspicious moles or small tumors. Incisional biopsies, on the other hand, are used for larger lesions where complete removal might not be possible or when a diagnosis needs to be confirmed before deciding on a treatment plan.
It sounds like the type of biopsy used really depends on the situation and what the doctor is looking for.
Precisely. It's all about choosing the most appropriate technique to get the most accurate and useful information.
This has been incredibly enlightening. It's amazing how much we can learn from studying cells, even in these tiny samples.
It is fascinating. It reminds us that even though we can't see them with the naked eye, our cells are constantly working, responding to their environment and telling a story. And it's our job with tools like biopsies and microscopes to listen to that story and understand what it means.
And sometimes, as you mentioned earlier, that story unfolds after death, during an autopsy. What exactly is involved in an autopsy, and why are they so important?
An autopsy, also known as a post-mortem examination, is a meticulous procedure conducted by a pathologist to determine the cause of death. It involves a thorough examination of the body, both externally and internally, looking for any abnormalities or injuries that might explain why the person died.
So it's like a final medical investigation seeking answers about what happened.
Exactly. And it's not just about closure for the family, although that's certainly an important aspect. Autopsies play a crucial role in advancing medical knowledge, improving public health, and even solving crimes.
That makes sense. By learning from every case, we can potentially prevent similar deaths in the future. What are some of the specific steps involved in an autopsy?
It's a systematic process that begins with a detailed external examination of the body, noting any scars, tattoos, injuries, or other markings. Then, the pathologist makes an incision to open the chest and abdomen, carefully examining the organs for any signs of disease or trauma.
So it's a very hands-on visual examination.
Absolutely. The pathologist looks for anything unusual like tumors, blood clots, fluid buildup, or organ damage. They might also take tissue samples for microscopic examination, just like with biopsies.
It sounds incredibly detailed. What else do they look for?
It's not just about looking. The pathologist weighs organs, measures dimensions, and documents everything meticulously. Fluids like blood and urine are collected for toxicological analysis to check for drugs, poisons, or other substances.
It's like gathering all the pieces of a puzzle to reconstruct the events leading up to death.
That's a great analogy, and sometimes the findings from an autopsy can be surprising, revealing underlying medical conditions that were not known during the person's life.
That's fascinating. It's a reminder that even in death, there's still so much we can learn about the human body.
Precisely. Autopsies are not just about the deceased. They're about expanding our understanding of health and disease for the benefit of the living.
It's amazing how much information can be gleaned from these post-mortem examinations. Okay, so we've talked a lot about anatomical pathology and how we study cellular injury, but we haven't yet touched on those lecture notes you have that are in Portuguese.
You're right. It'll be interesting to see what insights those notes offer. They provide a different cultural and linguistic perspective on cellular injury, which could be really illuminating.
I'm all for exploring different perspectives. What do you say we dive into those notes and see what we can learn?
Let's do it. It's time to put our translation skills to the test and see what these Portuguese notes reveal about cellular injury.
Okay, so we are tackling these Portuguese lecture notes on cellular injury, and one thing that jumped out at me is how they emphasize that injury happens when cells are overwhelmed by stress. They can't adapt or they encounter these harmful agents.
Yeah, that's a great observation. It really highlights that tipping point, that moment when the cell's defenses are breached and it can no longer maintain its normal state. And these notes go on to explain how this injury can actually worsen, progressing from a reversible stage to irreversible damage and finally cell death. There's even a diagram here illustrating this progression, showing how a normal cell under stress can adapt, recover, or succumb to the damage.
Yeah, it's like a flowchart for cellular injury.
Yeah.
I love how visuals can make these concepts so much clearer. It's like a universal language, even if the words are in Portuguese.
Absolutely. And it's interesting that they specifically mention different parts of the cell being affected, like those mitochondria we talked about earlier.
Right, those powerhouses are particularly vulnerable to stress. Yeah. What else do these notes highlight?
Well, they spend some time on reversible injury, focusing on those early stages or milder forms of damage where the cell still has a chance to recover.
So it's like catching the problem early before it becomes a full-blown disaster. What are some signs of reversible injury according to these notes?
They point to two main things: reduced energy production, specifically ATP depletion and cellular swelling, due to an imbalance of ions and water flowing into the cell.
It's like the cell is struggling to keep its energy levels up and maintain its internal balance. It really makes you appreciate how delicate those cellular processes are.
It really does. The notes even describe how this swelling looks under the microscope, saying that the cell appears enlarged and pale. They even use a specific term, hydropic change, to describe how the cytoplasm becomes watery and vacuolated.
So even at a microscopic level, you can see the cell struggling to cope with the stress.
Mhm.
What about irreversible injury? How do these notes address that?
They make it very clear that if the damage continues, the injury becomes irreversible and the cell will die. And this is where they introduce those two types of cell death we talked about earlier, necrosis and apoptosis.
Right, the messy collapse versus the
Mhm.
controlled demolition. They go into detail about the differences between the two.
They do. They describe necrosis as a pathological process that always indicates disease. The cell membrane breaks down, its contents spill out, and inflammation is triggered.
It's like the cell explodes, causing damage to its surroundings. Definitely not a good scenario.
Exactly. In contrast, they present apoptosis as a more controlled and even beneficial process. They actually call it cellular suicide, where the cell neatly dismantles itself from within without causing that messy inflammation.
It's incredible how the cell can orchestrate its own death in such a precise and organized way. Do these notes give any more insights into apoptosis?
They do. They talk about its role in normal development, how it removes unneeded cells during fetal development, and they emphasize how it eliminates damaged or infected cells, preventing them from harming the organism as a whole.
So, apoptosis isn't always a bad thing. Sometimes cell death is necessary for the body to function properly.
Exactly. It's a critical part of maintaining that delicate balance we talked about earlier. These notes really drive home that duality, showing how cell death can be both a destructive force and an essential part of life.
Yeah, it's a really powerful concept. Do these Portuguese notes cover the specific causes of cellular injury?
They do, and they categorize them in a very similar way to what we talked about earlier. They list oxygen deprivation, physical agents, chemical agents, infectious agents, immune reactions, genetic damage, nutritional imbalances, and aging as the main culprits.
It's reassuring to see that the fundamental causes of cellular injury are understood across different languages and cultures.
Absolutely. It highlights the universality of these scientific principles, and within each category, they provide specific examples. They mention ischemia as a major cause of oxygen deprivation and highlight the role of free radicals in causing cellular damage.
It's amazing how these notes bring together different concepts, connecting the dots between causes of injury and their impact on cells. Do they also talk about methods for studying cellular injury?
Yes, they do. They dedicate a good chunk of the notes to explaining anatomical pathology and its importance in diagnosing and understanding diseases by examining tissues. They even describe those different types of biopsies we discussed: exfoliative, fine needle aspiration, and punch biopsy. And they outline how tissue samples are prepped for examination under a microscope.
Wow, these notes are surprisingly comprehensive. It's like a crash course in cellular pathology.
It really is. And it highlights how intertwined these concepts are. Understanding what causes injury is key to interpreting the changes we see under a microscope, and those microscopic findings help guide us in diagnosing and treating diseases.
It reminds me that science is like a giant web of knowledge, and each new piece we learn adds to our understanding of the bigger picture. These Portuguese lecture notes have been a great addition to our deep dive. They offer a new perspective and reinforce those key principles we've been exploring.
I agree. They've definitely enriched our conversation and reminded us that knowledge transcends borders.
And that's what the deep dive is all about, exploring different sources, making connections, and gaining a deeper understanding of the world around us. Today, we took a deep dive into the world of cells, exploring their secrets, their vulnerabilities, and their amazing resilience. It's been quite a journey.
It has, and hopefully you, our listener, are leaving with a newfound appreciation for the trillions of tiny wonders working tirelessly inside you right now. Keep exploring, keep questioning, and keep diving deeper into the fascinating world of science. Until next time.